Hello and welcome to Brain Bytes. This is a series where we learn about the new and exciting research coming out of the Center for Brain Health here at the University of British Columbia. We're going to dive deep into new findings and then talk to the scientists that are behind the research.
Sounds look good.
There's a problem in neuroscience. Most of the research that's been conducted has been on male subjects. And this is true whether we use humans, rats, or even mice. Males are just used more frequently. And there are many reasons as to why this is, and some of them even make sense and are valid. But this has led to a problem, that we now assume that the brains of males and females are the same, and that the treatment that we give to males will work equally as well in females. Here lies the problem. Research has shown that this just isn't the case. Aarthi Gobinath is a neuroscientist here at the Center for Brain Health, and her work was trying to tackle how we understand depression in the brains of females. And her work in postpartum depression has some interesting results.
I'm here with Dr. Aarthi Gobinath.
Hi, how are you?
I'm doing very well. Glad you could be here today.
Yeah, happy to be here.
Let's get to it. What was your research question?
We wanted to know how would different types of antidepressants, meaning fluoxetine, more commonly known as Prozac, versus exercise, or the combination, how would those antidepressant regimens work in the case of postpartum depression?
And how did you go about studying that?
So we actually used a group of rat mothers and gave them stress hormones to induce depression-like behavior, and then applied different types of antidepressant regimens to see how that would affect their outcomes.
But you're probably wondering, how could they even test postpartum depression in rats in the first place? Well, it's actually not too difficult. The scientists injected stress hormones into the rats, and this would induce a depression-like behavior. They would then make sure to do this just after the rat moms gave birth to make sure that what we're seeing is postpartum depression.
So what were the results of the study? What did you find?
We found that each of these antidepressants had their own set of unique advantages. So fluoxetine had a positive effect on maternal care, exercise had a positive effect on depressive-like behavior, and the combination of these two antidepressants improved neurogenesis, or the production of new brain cells.
Weird that the third one needed a combination of both types of antidepressants.
Yeah.
Is that strange?
So it was unexpected, and it was unexpected because both these types of antidepressants alone, fluoxetine and exercise, have been shown to increase neurogenesis in male subjects. But what's interesting is that that wasn't true in the mothers. This actually, both the combination of these two antidepressants increased neurogenesis, which really highlights that we really need to better understand how the male brain is different from the female brain, and not just by comparing these two sexes, but also appreciating that there are different times in a woman's life where her brain may be responding differently to antidepressants.
So we're in the microscope lab, and Aarthi, what are we doing in here?
So we are going to be actually looking at the hippocampus tissue that was used in the study.
Okay.
So looking at that final measure, how do we measure neurogenesis in these rats? So over here you can see that this is the hippocampus. And this area here that's highlighted in purple, that's actually where new neurons or brain cells are being produced. And we can actually go ahead and quantify how many brain cells are being produced based on those different types of treatments that the moms were getting. What's really cool is that we can actually go up in closer and actually look at what those new neurons are, or where they are.
So you've zoomed in, and what are we looking at here?
So what we're looking at is actually new neurons that were just made over the course of that treatment that those rat mothers were getting. So this is actually, the darker stained cells are positive for a protein called double cortin. So this is something that's only expressed in neurons that have just been produced. So these are new-born cells that have been just made in the hippocampus.
So what does neurogenesis really mean to depression, though?
That's a great question, and that's one that actually researchers are still trying to understand. We think that neurogenesis may be lower in the case of depression, and that antidepressants may be actually bringing that back up, and maybe one of the ways antidepressants are working. But again, a lot of that work was done in males, and what we wanted to see is whether or not that's really true in the maternal brain.
What got you into this line of study?
Yeah, so I was really surprised. Before I began my PhD, I was surprised to learn that actually a lot of neuroscience research has been based on using male subjects. And that's based under the assumption that what's true for the male brain will be the same in the female brain. And now researchers are better understanding that actually that's not the case at all. There are a lot of things that are different about the way males and females present with different diseases, are at risk for different diseases, and of course how different treatments work in both of these sexes. And so for that reason, I wanted to really understand how depression would specifically affect women, because actually women are twice as likely to suffer from depression than men. And women are at an increased risk to suffer from depression during the course of motherhood, meaning over pregnancy and in the postpartum period.
Why do we think that there is this bias towards using male subjects?
So there is some struggle with communicating why it's important to study both sexes in research. So unfortunately, some researchers do think that it's twice as much work to study both the male and the female brain. From my point of view, I think we're getting twice as many answers by studying the male brain and the female brain. And then there's also an argument that some say that because females do fluctuate over the course of the menstrual cycle, which is in humans, or the estrous cycle, which is in rats, that it's difficult to control for that. But really, using statistics, we can actually analyze how these changes happen over the course of those fluctuations and better understand then how the disease or the disorder progresses over the course of the estrous cycle or the menstrual cycle.
That has completely shunned and removed the fact that, well, there could be differences in a female brain that should be addressed. And by not looking at those differences, we're preventing any kind of positive treatment that is needed.
Absolutely. I mean, right now in neuroscience, we're facing so many major diseases that need better treatment interventions, such as depression. But if we're only studying one sex, and when the other sex is suffering at twice as high of a rate, but we're not really using them, we're really hampering women's health by not studying them.
And if women suffer at twice as high a rate, clearly there must be some type of sex-based difference here.
Absolutely. So whenever we see a sex difference in some aspect of a disease, that's a major clue as to better understanding that disease. So when we see that twice as many women are suffering from depression than men, what that really says is that there's something about the female physiology, meaning something perhaps related to her hormones, that are actually influencing how depression is progressing.
Were you ever worried that the results of your study could be misinterpreted?
Right. So there have been ways that researchers studying sex differences in neuroscience, sometimes it gets misinterpreted or miscommunicated as some sort of biological basis for misogyny, or to really exploit that difference in a negative way, which is unfortunate because really as researchers, when we see that there's a sex difference, that's really exciting to us. That means there's an opportunity there to really innovate and find new opportunities and ways to discover something new about the disease. Maybe it is different in females, maybe it is different in males, and that's something that as researchers we can start to address.
Yeah, by acknowledging these differences, we're now able to treat people better.
Absolutely. For a really long time, not studying women or studying the female brain has really hampered what we know about women's health. When there's a similarity between the sexes or when there's a difference, studying both of those is an opportunity to improve the well-being of everybody, right?
Dr. Gobinath, thank you for telling me all about your research today.
Oh, thank you for the opportunity to share my research. And I also just want to give a shout out to the rest of the authors on this paper. This was certainly a team effort to pull off so much work. And I really want to thank my supervisor, Dr. Alyssa Galea, for all of her support and mentoring through this project.
That's great. If anyone wants to read your work, where can they go?
So it's actually just been recently published in the journal Neuropharmacology.
There will be links to Dr. Gobinath's paper in the description. Thank you for watching.